Good afternoon, everybody. Myself, Dr. Niranku Mondol, would like to represent a seminar on study on anti-cancer activity of sphingolipid isolated from attenuated Leishmania donovani. So, introduction. Now, in multicellular organisms, homeostasis is maintained by regulating the cell proliferation and cell death. From the early embryonic stage, cell division occurs in control wave. Any mistake of cell cycle checkpoint causes cluster of abnormal cells. The latter cluster of cell produces benign tumors. These benign tumors can easily be removed through the surgery. The benign tumor transforms into malignant tumor rapidly, and this transforms into cancer stem. Apoptosis play a vital role in deletion of neoplastic cells, in consequence preventing the progress of disease. The chemotherapeutic drugs which are applied for cancer treatment induces endogenous apoptosis. Now, apoptosis versus necrosis. So, what is called apoptotic cell death? What is called necrotic cell death? Necrosis is the one type of cell death which injury takes place at the cell membrane and cell swelling, membrane breakdown takes place, disintegration and inflammation ultimately occurs. But in apoptosis, DNA fragmentation takes place, decreases in cell volume, membrane bleeding, formation of apoptotic body also takes place. Now, structure of lipid, that is sphingolipid, is the important bioactive molecule. So, sphingolipids is a very important bioactive molecule that shares a basic structure. It can be divided into three parts: sphingolipid backbone, which is usually sphingosine fatty acids, and head group. The large number of different sphingolipids are made possibly by variation in sphingosine backbone, differences in fatty acid cells, length, degree of saturation, and degree of hydroxylation of fatty acids, and variation of head groups. Now, next, bioactive sphingolipid isolated from attenuated Leishmanial donovani have a special attention in case of cancer therapy. So, chemotherapeutic agents create many pathophysiological problems. It has been found that the synthetically produced drugs have been many long-term effects on human body system. These synthetically produced drugs have many side effects on normal and healthy cells. Lipids, mainly isolated from the attenuated Leishmania, induces apoptosis in both human and mouse melanoma cells, have been reported earlier, that is Roth et al. 2006 and Dr. Bhadra groups. So, sphingolipids destruction isolated from this parasite have a special interest on cancer therapy. Even sphingolipids also show immunomodulatory effect on rheumatoid arthritis patients, synovial cell McCoy. Hence, bioactive molecules have important role as immunomodifier without any side effect. Now, objective of this work. Main objective of this work is given below. Isolation of sphingolipid destruction from attenuated Leishmania donovani comestible. Separation of major components of sphingolipid destruction. Each lipid fractions are collected and were named LSPL1, LSPL2, LSPL2 and LSPL3. Next, justification of antiproliferative activity on several human cancer cells like HeLa, FG2, SKML5, MIA-PACA2, HCT116, K375, mouse melanoma cells and other human cells. Verification of LSPL1 and LSPL3 sensitized cells apoptosis through phase contrast and infrafluorescence microscopic study of these malignant cells, HeLa and FG2 cells. Special attention was given on apoptotic activity by LSPL1 two cells HeLa and K562. Investigation of detailed mechanism of apoptosis initiated by sphingolipid destruction isolated from attenuated Leishmania. Now, result and discussion. Attenuated Leishmania donovani comestible grows in raised modified medium. Thin layer chromatogram of sphingolipid extraction obtained from salicylic acid column chromatography using chloroform methanol ratio 2:1, lane on is to 6, lane 2, lane 3, and stimulated of bash on and bash 2. LSPL on, LSPL 2, LSPL 3 was also detected by benzene spray. The spots are dissolved with chloroform-methanol and water and visualized by benzene spray and hydro spray. Now, effects of LSPL on cell viability. Here the figure shows that the LSPL on cell viability in different cells U937 HEP2, A375 HeLa A562 and THP1. Dose-dependent manner results are expressed as mean, plus and minus, standard error means of 3 independent experiments.